Thank you so much, everyone. Now we are going to move to the short pitches. I don't know, Hussein, are you ready to tackle the floor?
Hello, everyone. It's a pleasure spending the day with you, by the way. I'm Hussein. I'm the co-founder and CEO of Red Crow. Red Crow is an online platform that presents a risk mitigation solution to those living and operating in volatile political environments. I come from Palestine. I live in Jerusalem. So born and raised in the West Bank, and doing my bachelor studies there in the peak of the violence period in 2005, what we call the uprising. It was very challenging for a lot of people, especially youngsters, to take simple decisions, such as moving from point A to point B, heading to the university, or worst case, delivering someone to the hospital for some reason. Mostly because living in a conflict zone, usually you face things like checkpoints that might delay you for hours, or you face confrontations between two parties that weren't expected. So this made me a bit interested in security and in the trends we can see within this chaos we're living in. Because we're always, when we ask about the issues, it's always security related. So I moved to the UK. I did my master's degree in Middle East politics, specializing in security. I thought more research was required, so I was an intern in security companies, and I worked for a few years in the private security sector, and did my second master's degree in homeland security. During that period, I was looking further about intelligence and trend analysis, and unfortunately what we were living through in the West Bank was reflected all over the Middle East, but by that time, because of what we refer to as the Arab Spring. Non-state actors taking over, economies collapsing, and further. So decided to start working on this more seriously, met with my co-founder, Layla. She's an expert in social media and e-marketing, and we launched our online platform that aggregates data from social media and open sources, including the public, governmental sources, RSS for people like Red Cross and UN, and decides if this data is correct or not, and allocates current and potential physical security threats. For example, if a protest is being mobilized, we allocate it, we measure how big it will be, and the effect based on the geopolitics it's occurring in, and so on. So we concentrated too much on the B2C concept because we wanted to serve the individuals operating on the ground, the people living in those areas. After 2 years of serving people like Amnesty International, European Union, USAID, and others who operate in volatile environments, we discovered that actually what we have is more valuable. The culminated data, the data we have, can actually produce much more value. So we started looking into trends within the data and analyze the ripple effects. Today we are developing our big data infrastructure on the cloud. We're working on natural language processing in Arabic, as it's a challenging part. And we're building our forecasting algorithm in order to be able to look at tomorrow and forecast based on the historical data we have and the trends we understood out of security interactions between the public and what's happening, and be able to see what might happen on a country threat level. An example which is very clearly important when we look at the Egyptian case, for example, when people for hours were saying the regime would collapse, it wouldn't. Should we evacuate many people or not? Shall we send aid or not? Currencies would change. So we're giving now a numerical analysis of threat levels and where they might be tomorrow by looking at the data we have been collecting yesterday. Today we are operating in Palestine, Israel, Egypt, and Jordan, and raising investment in order to cover MENA region in 18 months. Thank you.
Perfect timing. So our second science slammer will be Mark. Mark is originally from the UK, is that right? Okay. I remember well. He's based here at NIH. And I will pass the microphone to you. Thank you, Mark.
Thanks, Victoria. I'll stand over here because I don't have a slide advancer yet. I use this one. So I've been working in the lab of Barry Richmond at the National Institute of Mental Health, and we've been trying to modify monkey behavior with molecular tools. So it's a big change of track from the previous talk. In a general sense, what we're looking to do is manipulate the brain. So in the case of human neuropsychiatric disorders, we reason that something goes awry in a specific part of the brain or in a given brain circuit that leads to neuropsychiatric disorders, anything from Alzheimer's, schizophrenia, depression, OCD, etc. It's one of the major unmet needs in the biomedical field at this time. So the technology we've been developing, what we'd like to do with it is to be able to pinpoint a specific part of the brain and to manipulate it in some way, to change how it's functioning from the norm, either to increase the activity or to reduce it. And the data I'm going to show briefly here involves a construct we've been using to reduce activity in specific parts of the brain. So this slide gets a little bit technical, but I'll do my best to explain it. What we're doing is expressing engineered receptors in the monkey brain. So we've taken, or molecular biologists have taken endogenous receptors from the brain. They've modified them so that they're not bound by any neurochemicals that exist in the brain. And instead, they're bound by a drug that we can administer systemically, either orally or intramuscularly, for example. What they do is to package the DNA of that receptor into a virus. That virus, when it infects a cell, these are, I should emphasize, these are non-replicating viruses, so they're not pathogenic. They discharge their cargo into a cell, and then they die, and they're no longer potent. The cargo that we load into them is this engineered receptor. So this receptor has been modified so that it only, as I mentioned earlier, it will only be activated by an exogenous ligand, not by anything naturally present in the body. And this particular type of receptor, when we activate it, it will silence the brain cells. So whichever region of the brain we've injected it into, all the neurons that have been bound by that virus and are now expressing our receptor of interest, their activity will be lowered. So they won't function as they did before. There'll be a block in the pathway. So what we've done in monkeys is to inject this virus into a specific region of the brain. Here I'm visualizing it with magnetic resonance imaging, MRI. So most of you have probably heard of that acronym. It's commonly used in humans. It's a non-invasive technique. So here you can see the region in which we've injected the virus. And subsequently, we want to know that we've expressed the virus, that not only did we inject the fluid in there, the vehicle containing the virus, we want to know the receptors are now being expressed in the cells so that they can do something functional. And the way we do that is with another non-invasive technique. So this is an image taken with PET imaging, positron emission tomography. This is another non-invasive technique used in humans. And the rough overview is that you can take a drug, conjugate it with a radioligand, in this case carbon-11, and then you inject that systemically. It will bind to any of our receptors that are expressing. So you can see here the comparison between the two images. We're getting the nice bright red signal in the PET in the same area we can see we injected in the MRI. And they overlay nicely. And I didn't get to the behavior slide, but I can show you it modifies behavior. Here there's no drug on board. Here we've put the drug on board, and the monkey no longer distinguishes between the different values of reward that the monkey has available. I'm sorry, I didn't have time to practice in advance, so hence the hopeless timekeeping.
Can I invite Karen to the floor? Fantastic. Here you go.
Thank you. So I also don't even have any slides. I'm so unprepared because I just decided 10 minutes ago. But I'm an immunologist, so I study chronic inflammatory diseases. I think there's not one person in this room who hasn't encountered the immune system, whether it be from mild hay fever to an itchy hive, which I have at the minute from a mosquito bite, to more debilitating disorders such as rheumatoid arthritis, ulcerative colitis, multiple sclerosis, for example. And chronic inflammatory disorders have actually reached epidemic proportions at this stage. Autoimmune diseases in the US alone, for example, have risen 3 to 10 times in the last 40 years. Diabetes, type 2 diabetes, has doubled in the last decade, and cancer worldwide has risen 20%. So the area that I'm interested in is a certain type of cell called a T cell, which is very important for eradicating tumors. And now, if you've ever had a cold or a virus, you will have encountered your T cells because that's what causes the glandular swelling that you feel. T cells then become switched off as you clear your virus, but they are long-lived and that they protect you throughout your life. Now, the problem with cancer is that they are actually, tumor cells are one of the smartest immunologists around, and they have figured out how to constantly turn off your T cells into this inhibitory state. There's a few molecules that have been discovered so far, but they're in the family called inhibitory molecules. And tumor cells are very clever at turning on these inhibitory molecules in T cells, which prevents the eradication of tumor in the vast majority of patients who are unfortunate enough to have cancer. So what we are discovered in my lab, I work with a number of people who have pioneered the discovery of these molecules and these treatments, is that you can use antibodies to target these molecules. And the efficacious quality of these is that chemotherapy, which is just a kind of a blanket treatment and kills, it's cytotoxic, meaning it kills all cells. If you use antibodies to inhibitory molecules, it specifically switches off the inhibitory molecules on these cells so that you can re-educate your immune system and that you can eradicate the tumor within yourself. It's not toxic, and it's more specific, and it's long-lived. So the inhibitory molecules which have been identified so far are actually already in use in patients, but PD-1 is one of them and TIM-3 is another. They only work in 30% of patients because we don't understand the full repertoire of inhibitory molecules. What I do is to further investigate the full plethora of inhibitory molecules and how they can be used in combinational therapies to target as many patients as possible in the most efficacious way, and that also maintains their quality of life rather than chemotherapies and so on, which of course have many side effects, which some of you are maybe aware of. And so that is it. I think. So thank you.
Thank you so much, Karen. Thank you, Karen, so much for standing up in front of all of us. And now we are going to continue with Pedro.
And I'm an undergraduate student at Georgia Tech, and I'm here as part of European Horizons. So I'm going to speak to you for a few minutes about a research paper I wrote. So it's called Galileo, European Collaboration for Space, and it shares several examples of European technical collaboration. So the first is obviously Galileo, the global navigation satellite system that the EU is creating via the European Space Agency and the European GNSS Agency. And it talks about how it has brought the EU and European industry to the forefront of space. And it starts off with really a historical description, and then it goes to technical and financial analysis. And it really highlights the intergovernmental and the supranational component of this project. The second example of European collaboration that this discusses is the Global System for Mobile Communication, which many of you know as GSM, which is the biggest European technical collaboration success story. And it's called European Collaboration for Space Galileo. So that's my short summary on this research paper that I've made.
Thank you. Or not, we are going to continue with Michael. The floor is yours.
Thank you. Hello, everybody. Like others here today, I am a postdoc at the National Institutes of Health. I don't have any slides, I don't have any notes, but that's cool because I just joined the NIH half a year ago, so I don't have that many results to show you. But I am a structural biologist. Structural biologists use different methods to look at tiny molecules in detail and use their findings to facilitate drug development for one disease or another. And I came to the NIH from Tübingen in Germany to learn electron microscopy. Electron microscopy has made some incredible advances in recent years that allow us to look at tiny molecules in the nanometer range in really, really great detail, and it's become increasingly easy to use that. And I'm going to learn electron microscopy by looking at viruses and looking at different stages in the life cycles of viruses, particularly herpes virus. So like children, like humans, as new viruses get made in a cell, they undergo certain changes and then they leave the cell as a mature virus. And what we in my lab at NIH have is a few really cool mutants of viruses that have certain proteins switched off, so they are trapped in one particular stage of their development, basically being trapped in puberty forever. And we can isolate these viruses from cell culture, purify their particles, and look at these particles in detail. And what we hope to find out is what the structural changes are going to be as these viruses go from the nucleus of the cell through the nucleus into the main part of the cell, and how they escape, for example, how they package the DNA, which makes them really infectious. And with electron microscopy, we hope to find really high resolution structures that will help us combat this really, really pesky virus that is at best a nuisance in our daily lives, but can become really, really critical when it goes into the brain. So I'm looking forward to some really incredible years at NIH and some great findings. So stay tuned. Thank you very much for allowing me to speak here.
This also was a super fast pitch. So my alarm is going to ring in about a minute. But after it does, we are going to welcome Ulrike on the stage. Ulrike, welcome.
Thank you very much. I hope you can all hear me very well. So, just starting out with where are we right now? So we are right now at Google, one of the biggest companies, kind of software companies, I guess, worldwide. But there are also lots of other companies out there, and those companies usually they produce products, right? And in order to make sure that a product can be produced, you have to make sure that the assembly line from the idea to the production, quality management, and so on and so forth is actually guaranteed, such that a product comes out in a nice way. And it's kind of similar with cells per se, because cells, or in particular the nuclei in a cell, is also actually a huge factory or company which kind of produces specific proteins, and those proteins have then also a specific role within the cell. So, but the thing is that like in a company where certain paths within the production scheme can go wrong, the same thing is actually true for nuclei, or the same thing is true for DNA, meaning under certain circumstances, certain genomic diseases can occur. And in order to understand actually where the bottleneck or where the errors kind of occur, what you would normally do in a normal factory, you would just observe the different kind of points, and then you would figure out, okay, here's something wrong, and we have to change that. But in a cell, it's a little bit more tricky, because the problem with the cell is it's not kind of as easy to kind of observe where errors occur, because in principle, we don't deal with the same length scales as we do in a company, but everything is much more compact. So in principle, we have a DNA thread, which is almost 3 meters long, and this DNA thread is packed in a nuclei of a size of approximately 2 up to 3 micrometers. And that means that you really have a very dense environment there. So it's kind of from the outside very hard to kind of figure out how actually gene expression works, and how to, for example, also to correct for an error. So, and what I do, so my background is in physics. What I do during my postdoctoral research at the NIH, I'm building a new advanced light microscope to actually figure out how gene expression within the nuclei works. And that in a way that I can really kind of figure out whether, if that's kind of in a direct way or more in an indirect way, how different sequences within the genome actually interact. And as I said, since the length scale is really very teeny tiny, it's pretty much kind of challenging, because with a normal light microscope, you cannot really do that. But you need a bit more advanced technology, and therefore, in my research, it's kind of very crucial to bring together biology and physics also at the same time. Okay, thank you very much for your attention.
It was 3.00. Wow, that was perfect. Okay, so we are going to have a last pitcher, last Science Slammer. Matthew, the floor is yours.
Thank you. The C. elegans worm has 302 neurons in the adult stage. And many neuroscientists use this worm to understand the basic functionality of the neuronal system. And they do that by imaging the worm in real time using fluorescent markers. However, it is very challenging to label those neurons, and it takes a lot of time. What we want to do is use a community effort to allow scientists to contribute their labeled data, which they've worked very hard on, to an open source system, which will then update itself, build models of neuron labels, and give back to the community. When the system gets unlabeled data, it will make predictions. We want to use a probabilistic way, using machine learning, an inference model, which will learn from the labeled data and give predictions of what the possible labels for each neuron in an unlabeled data set could be. That's it.
Voila, voila. We came to a conclusion, all of us together, including the judge. We have the name in the envelope. Again, I would like to thank all the contestants to come up, especially the brave ones, and all of you for listening. So I'll pass the microphone to the main judge, Yolanda, who's going to announce the winner.
Okay, so I am very pleased and proud to announce that the winner of the Science Slam is Hussein. Congratulations and thank you so much. And would you like to say a few words?
I've been speaking for a while, but it was really a pleasant meeting you all. And thank you for your efforts, your great efforts, and looking forward to stay part of this community. So please keep us posted. Thank you.
Thank you.